Welcome to this explainer. I am absolutely thrilled to have you here today. We are going to embark on a really fascinating journey into the hidden mechanics of plant reproduction. We're calling this Bloom to Bite, the hidden journey of fruits and seeds, and honestly, it is a world of microscopic miracles happening all around us every single day. Let's dive right in.
So have you ever actually stopped to ask yourself, why do plants even produce fruits and seeds? I mean, we eat them all the time, right? But these delicious little treats aren't just there for our culinary benefit. They are incredibly clever biological vessels designed by nature for a very specific survival-driven purpose. Okay, here's our roadmap for today. First, we'll look at the anatomy of a flower. Second, the pollination process. Third, nature's clever pollinating agents. Fourth, double fertilization explained. And finally, from flowers to plates.
Part 1. Anatomy of a flower, setting the botanical stage.
To really understand the food we eat, we first have to meet the key players. What we have here is the gynoecium. That's the female reproductive part of a flower. Notice the three main structures. Right up at the very top, you've got the stigma, which is essentially the landing pad that catches pollen grains. Just below that is the style, this sort of elongated, slender pathway. And all the way down at the bottom, that bulged part, that's the ovary. Tucked safely inside that protective ovarian cavity is the placenta, which holds all the vital elements for reproduction.
Now, zooming right into the ovule or megasporangium, attached to that placenta, look at how incredibly well protected it is. You can see these layers wrapping around it. Those are called integuments. But they intentionally leave one tiny, absolutely crucial opening right at the top, called the micropyle. And sitting inside those protective layers is the nucellus, which contains the reserved food, just waiting for the whole process to kick off.
Getting even closer, we reach the mature female gametophyte, or the embryo sac. This microscopic structure is honestly mind-blowing. At maturity, it's 7-celled and 8-nucleated. Down by that micropylar opening we just talked about, we have the egg apparatus, featuring the vital egg cell and 2 synergids. Those synergids have these special thickenings called the filiform apparatus. You can think of it like a biological runway guide leading pollen tubes exactly to their target. And right in the middle sits the central cell, holding 2 polar nuclei. All of these complex anatomical structures are sitting there, patiently waiting for one defining event.
Section 2. The pollination process. Transferring the pollen.
So when it comes to getting pollinated, plants basically rely on 2 overarching strategies: self-pollination and cross-pollination. Self-pollination, or autogamy, is basically the safe bet. It preserves parental traits and guarantees continuity, which is great if pollen is scarce. But it's genetically a bit stagnant. It runs the risk of inbreeding depression, which gives us smaller, weaker seeds without any cool new varieties. On the flip side, cross-pollination is a huge gamble. It relies entirely on outside agents and literally wastes a ton of pollen in the wind or on passing insects, but the payoff is massive. It combines desirable traits, creating hybrid vigor and seeds with way better germinating capacity. And here is that gamble paying off beautifully. This is cross-pollination in action, taking pollen from the anther of one flower and depositing it onto the stigma of a completely different flower of the same species. Nature actually actively encourages this genetic mixing. In fact, plants use some brilliant contrivances, like dichogamy, where they mature their male and female parts at totally different times, just to force cross-pollination to happen. But if a plant can't uproot itself to go find a mate, how exactly does it pull this off? Which brings us to Section 3. Nature's clever pollinating agents, the heists orchestrated by nature. To actually pull off these tiny heists, plants will manipulate wind, water, and animals to do all the heavy lifting. Let's look at a super fascinating example of insect pollination, or entomophily, in the salvia plant. It is a highly precise 3-step trap. Step 1. A bee enters the flower looking for some nectar and bumps right into a sterile anther lobe. Step 2. This triggers a mechanism where the stamen bends down with a sudden jerk. And step 3, boom, pollen is sprinkled all over the back of the unsuspecting bee.
Just look at this incredible lever mechanism go. Watch how the fertile anther lobe perfectly strikes the insect's back the second it pushes that sterile lower lobe. The pollen is now perfectly positioned on the bee, so when it buzzes off to visit the next flower, one that has a ripe protruding stigma, the pollen will effortlessly brush right off. It is literally a mechanical trap.
Now let's switch gears and see how this plays out in a completely different environment. Water. Pollination by water, or hydrophily, is actually pretty rare, but the Vallisneria plant has it completely mastered. Step 1. When they ripen, the male flowers physically detach from the plant and float right up to the surface like tiny little boats. Step 2, they drift along and cluster around the female flowers that have reached the surface on these long stalks. Step 3, after they release their pollen onto the female stigma, the stalk of the female flower coils up like a tight spring, pulling the fertilized flower back safely underwater to develop. Nature is just wildly inventive.
And plants will honestly go to whatever extremes are necessary. Take ornithophyly, or bird pollination. These plants grow massive, brilliantly colored flowers packed with nectar just to support the heavy weight and appetite of a visiting bird. But then look at anemophily, or wind pollination. Plants like maize totally ditch the colors and the nectar. Instead, they produce super light, non-sticky pollen and hang these massive feathery stigmas out in the open to catch the breeze blowing across the fields. Now, whether it's by bee, boat, bird, or breeze, once that pollen successfully lands on the correct stigma, we reach the defining climax of our story.
Section 4. Double fertilization explained. The scientific climax.
Okay, this exact biological magic trick is completely unique to flowering plants. Double fertilization is a process that requires two separate fusions to succeed. First, one male gamete fuses with the egg, a process called syngamy, to form a zygote. But then, a second male gamete fuses with those two polar nuclei we saw earlier. This one is called triple fusion, which forms the endosperm.
So, tracing that final journey, the pollen grain germinates way up there on the stigma. It then grows a long pollen tube all the way down the style, navigating straight to the ovary. It enters the ovule through that tiny micropyle opening, guided smoothly by the filiform apparatus into the synergids. Once it's inside, it releases its two male gametes to execute that spectacular double fusion.
And here is the immediate aftermath inside the embryo sac. That zygote we just formed is preparing to develop into the actual plant embryo. Meanwhile, that central cell has now become the primary endosperm cell. Now, why does this matter? Because this endosperm is going to serve as the absolutely crucial food supply to constantly nourish that developing embryo.
Which leads us nicely to Section 5, From Flowers to Plates, the Edible Conclusion.
So how exactly do these microscopic cellular fusions end up on our dinner tables? Well, after fertilization, the other parts of the flower just fall away, and the ovary itself ripens into a fruit. Take a look at the cross-section of this mango. The fruit wall, or pericarp, develops directly from the ovary wall. That sweet, fleshy part we love to eat, the mesocarp, is just a ripened botanical structure meant to attract animals. The epicarp is the outer skin, and that hard innermost endocarp, that is there to aggressively protect what is hidden inside.
But what's hiding inside that hard pit? The seed, of course. That mature ovule we've been tracking this whole time is now wrapped in a hard seed coat made of two layers, the testa and tegmen. Inside sits the axis, which holds a radicle, that's the future root, and a plumule, the future shoot. The seed is packed tightly with nutrients in the cotyledons, basically just waiting for the exact right environmental conditions to germinate and start the whole extraordinary cycle all over again.
Think about this for a second. Human civilization literally runs on these biological marvels. They make up our staple diets. We've got pulses, like peas and lentils. Those are protein-rich seeds from the Leguminosae family grown in pods. We have grains, which are hard, dry seeds mostly from the grass family, like wheat and rice. And we have millets, which are coarser grains with high protein and smaller seeds like jowar and bajra.
To really put this into perspective, just look at world grain production. A staggering 85% of all total global grain production relies on just 6 types of these mature ovules: maize, rice, wheat, barley, oats, and sorghum. That's enough to feed billions. The remaining 15% covers all other grains combined. We literally rely on the successful double fertilization of these specific plants for our survival.
Which leaves us with this final thought. Next time you bite into a crisp apple, or slice a juicy mango, or even just sit down to a simple bowl of rice, ask yourself, whose incredible botanical journey are you actually tasting? From a tiny pollen grain hitching a ride on a bee's back, to a complex double fusion deep inside an ovule, right up to the protective vessel of a sweet fruit, it is a totally hidden world of biological heists and microscopic miracles happening right in front of us. Thank you so much for joining me on this explainer, and hey, stay curious.